Practical 3 is one of the prescribed pracs in the IB course, and it's the enzyme design prac where you have to choose your variables out of temperature, substrate concentration, and pH. And then really it's great practice for the IA because you need to collect some data. You should be looking at testing at least 5 increments. So say, for example, in this spreadsheet I've set up here, I've got 5 different temperature values. And then you should be looking at taking at least 5 replicates. They're the trials, so that you can generate a mean and standard deviation and plot a graph that has a range of data and some error bars to show the variability in your trials. You should also be looking at part of good experimental design here. If you're looking at the effect of an enzyme, you should be running a control, which is an experiment where you're using distilled water in place of the enzyme. And reporting in all that data. So here I've got just some mock results to help you if you're having a bit of trouble with your Excel spreadsheets and doing your graph. So here for starters, I've sort of half set it up, but I thought I'll show you a couple of little things in case you've forgotten from our last practice, some of those tips and tricks with using Excel. So for example, neatening up this graph here, this temperature bit, I can highlight, click and drag these cells here, and there's a button up here called merge and center. So you need to be on the home tab at the top for that to be visible, or you can go for more settings in the alignment. You can open this dialog box here, but just click on this button, and then also this one here, this ABC with the arrow is called wrap text, and that breaks it over several lines if it doesn't fit inside the cell. So there we've got that one there, and you might also like to just drag it out a bit so that it doesn't break a word in half. Now the units I've got here, you should report like degrees Celsius here for temperature is what we measured. If you're not sure where that degrees symbol is, you can go to the insert tab and you'll find symbols over on the right here. So if you insert symbol, you can see some of the ones that I use quite regularly pop up there. A lot of the basic fonts used by Microsoft have a lot of the mathematical symbols. So if you scroll down, you'll find a little degree symbol, you'll find the plus minus value, which we use for uncertainties of equipment. And you know, other things like delta, for example, is often used in science to represent change, or here we've got sum of as well. So I've got that in there. I've got my units. I have to have units in the headings of your table. Don't put them in with the actual values, with the numbers, because Excel won't be able to calculate values if you've put other text in there. So leave your units there in their headings, and you should try to estimate the uncertainty. So that's that plus or minus value. So with temperature, you might be looking at half the smallest increment that you can read on the thermometer, or here I've just rounded that up to 1. Because that was the temperature of our water baths that we did our experiment in, and I don't think it's really accurate to suggest that they would have been so precisely held at exactly that temperature. I think it's reasonable to suggest that the water baths might have fluctuated within about a degree either side of what they were set to. So that is a reasonable estimation, and you can always justify that in the written part of your results as well. So that's here for temperature. Here, this is one where I'm saying, for example, we tested catalase with hydrogen peroxide and saw oxygen given off, and you might have collected that as a volume. So say it was the setup where you put the flask with the delivery tube underwater and you had a measuring cylinder filled with water, and then the volume of oxygen gas that displaced the water could be measured off. So that one here, again, we might just neaten that up. Go back to the home tab, merge and center. Then I've got my trials of catalase, and I've got my control, which is my no enzyme one. So again, I'm just doing a little bit of formatting to make that look a bit neater, and I've got my 5 trials here. You can put the mean and standard deviation in the same table if you want to, or you can put it separately, as long as you make it clear that you've got the data you originally collected and you've done some processing to show how you've got the values you've plotted on your graph as well. You might also like here on this one, I've already got all borders, but if you haven't, just have a look down this menu, and that just puts lines around your table. Then when you copy and paste it into Word, it looks nice as a table. Okay, so here that's my sort of raw data, the data that you would have collected. Now, if I want to report, my research question is actually talking about the rate of enzyme activity, then I need to turn my values into a rate instead of just a volume. So if it took 30 seconds in this example to collect these volumes of gas, I'm going to divide all these values by 30 so that I get a value in centimeters cubed per second. Now, as you can see here, I'm just going to go up into this box. I haven't formatted this yet, so I can show you. Oxygen, the O2, you need to highlight it. Just use the arrow keys and shift. And then here in the font setting, now you can't do this inside one of the cells. You have to go up here in this formula bar to be able to like manipulate your font. So go to this font and subscript. And here, for example, centimeters cubed. Now, centimeters cubed is the SI unit for volume rather than milliliters. It's preferred for scientific documents, but a centimeter cubed is the same as a mil. So font, go to this little font settings tab to give you more options, and superscript to go up high. Now, when we're writing per second, I've already used this / here to indicate my unit, so I don't want to put another one. It's also convention to just write them with indices, like powers. If we would have had per, like divided by, you just use your index laws to make it negative. So here I'm going to do this per second. I can use this -1. So again, superscript for that one. So now down here, this is looking a lot better. I can copy and paste my temperature values here. And then I'm still going to have my trials. Now you can do your control one as well if you like. It's probably not as essential because you've seen that the values are really quite small, and it's more for your ability to draw a comparison with your results, and it's not going to be what you plot, but it's up to you. If you want to continue, if you think you've got big enough results here for your control that you want to convert that to a rate, you could do that as well. But I'm just going to stick to my trials here with catalase. So now I want to do a bit of processing and show you what that would look like. Here I'll just put mean and standard deviation. Now I'm just going to merge those cells and wrap text to make those a little bit neater. If you want to as well, if you don't like it sitting at the bottom of the cell here, up here you've got the option to put it in the middle, the alignment, change those sorts of things if you want to. If you prefer, you might want all your values, for example, centered. Often when you see IB exams, that's just their style preference. They put them in the middle, it doesn't matter. But I'll just do that. Might as well be consistent. I've sort of copied this layout from an exam question, the way they would do it. Okay, so here I've got my temperature. Now I'm doing it as a rate. So I need to use a formula. Thankfully, this is all the columns are aligned with the information above, which makes it really easy to copy a formula across. So I'm going to type equals. That's how Excel knows it's going to do some maths for me. Here I'm going to press click on the box with the first lot of information for the first trial at 32 degrees. Now that was my volume of oxygen collected in 30 seconds. So per second, I just have to divide that value by 30. That's pretty easy. So what I can do now is I can click on that answer that it's generated, move my cursor. At the moment, it's a white cross. Move my cursor, the mouse, over to the bottom right until it turns into a solid black cross. Now I can click the mouse and just hold it down and drag, and it will copy the formula down that column. If I want to as well, I can highlight those 5 values. Again, black cross, click and drag across, and it fills in all the rows. Now, because Excel was the way it's designed, when you copy a formula, and then if you look up here at the formula bar, you can go back to the information and correct it if it's done something wrong. But it's taken information from cell B4. So if I've moved that formula across one, it automatically adjusts to C4. Or if I go down, here it goes to C5. So it's done all that for you, which is handy as long as your second table has the same width and things like that. so that it copies the right thing, it adjusts correctly. Now here my significant figures are a mess. For the most part here I've got two significant figures. The volume we've just read off the measuring cylinder to a whole mil because it's pretty hard to go smaller than that, or depending on how precise the instrument was. And my temperature values have just gone to two significant figures. So I should be consistent here with these results. Now these tabs up here will move the decimal places to either increase or decrease them. So I've just decreased them all down, but they've gone too far. So here, if I increase them back up, now they have two significant figures, except for these last ones. These placeholding zeros out the front don't count. So I'm going to increase just that row. So it won't necessarily be the same number of decimal places. It's got to be the same number of significant figures. So that means the zeros, only if they're in between two other digits do they count. So, or if they're after a decimal point. So this one here, 2.0, that's two significant figures. But here, this zero at the front, just a placeholder. 0.17, that's got two significant figures. Okay, here for my mean. So that's your average. See, that's the formula Excel uses, equals. When you type the word average, you have to put an open bracket in, then highlight those values you want to take the average of. So click and drag across those 5, close the brackets and press enter. And we can copy that formula down as well. So click on the cell, move across to the black cursor, click and drag, and we have an average value for all of our temperatures. This one, again, I'm just going to increase by 1 decimal place so it has two significant figures. Standard deviation. Now this gives you an indication of the spread of data here, so how close all your trials are to your mean value. So we type equals STDEV brackets. And it's sufficient when explaining this in your IA or prac write-up to say what formula you used in Excel, or just explain a little bit about what it's showing. You don't actually have to include the mathematical process, as long as you indicate that you use this formula in Excel. What it's actually doing though is taking each value and looking at the difference between that and the mean, and it's squaring it to make sure the difference is positive, so it can add them all together, and then it takes into account how big your sample size is as well. I'm sure you've looked at it in maths. So click and drag. Go across your 5 values. Be careful not to accidentally go too far and include your mean, because that will reduce your standard deviation. It will think that you've got an extra trial that's bang on exactly the same as your mean. So just calculate those first 5. Close the brackets, press enter, copy and paste. Neaten up the significant figures. So again, you're going to have to do some of them individually so that they've all got two significant figures. Okay, we're time to plot our graph. Both these tables, by the way, you would put into your Word document to show the raw data you collected and the processing that's occurred. You should put a heading at the top of each one. So you would put like the first one, table 1, give an informative, descriptive heading. Table 2 for this one here. Convention dictates that we put headings at the top of tables, and you would call them like table 2 and then put your heading in here. I'll do that one. Okay, then we make our graph. Now, I don't suggest trying to include headings when you first plot your graph. Just select the data, because it gets messy with all these cells and it gets a bit confused. If you've got your independent variable to the left or at the it should be able to identify that and plot it on the x-axis. So click and highlight your temperature values, then let go with your mouse, hold then the control button down so that you can click and drag something that's not right next to it. If you don't press the control button, and you just click and then try to click over here, it's like lets go of the first lot of data. Or if you click across all of it, you get too much data. So you have to click this first column, let go of the mouse, then press the control key and click these ones down Okay, and then you can let go of the mouse and the control key and go up to insert. And here we've got some charts. You can go down and choose which one you like. You might just want to do the points for now, or kind of a dot to dot, or a smooth curve can be okay for this sort of one as well. Sometimes it puts misleading like little wiggles on there, so you might actually want to just go back to a plain kind of dot to dot one. But make sure the data points are actually on there. So here's this one. Now we're not going to do a trend line for this one. It's unsuitable for this type of enzyme graph. A trend line is a line of best fit that's mathematically calculated. That's much more suitable if you've got perhaps a lot of data and you've got a trend that's very definitely linear or quadratic or exponential in nature that you can fit a mathematical trend line. For this kind of data, you can't. Okay, now I've already cheated and put like a little caption at the bottom here. So I've called that figure 1. So we would put that down the bottom underneath the graph, like the effective temperature on enzyme activity in catalase as measured by the initial rate, the first 30 seconds, of oxygen volume loss from hydrogen peroxide decomposition. So I've given it a really informative caption down the bottom. I don't need this chart title that it's going to generate for me here inside the box. So click on the graph, then click on this bit here so that a box comes up around it, and you can just press the delete button and it will go away. So I don't want that. I do want a few other things in my graph though. So I'm just going to scroll across a little bit. If I highlight this, plus, now I want some axis titles. So I'm going to tick that box. And I want some grid lines here I've already got, but I'm going to go to this little triangle here's arrow, and I'm going to put some minor ones in there so it looks more like graph paper. It's easier to read. And I'll do a few other things in a minute. Let's just tidy those up for now. So here, axis title, this one is my volume, my rate. And in fact, I'm just going to take the information from my table, copy that, and paste it. Because when you're writing a scientific article here or report, it's not the time to get fancy or get out a thesaurus. You actually just want to be really consistent with your terminology and be really precise about what you're saying. So here we go. So I'm just tidying up these things, moving my cursor to the spots where I need to fix my font, because unfortunately it doesn't copy across the formatting of my subscript and superscript letters here. Okay, here, this one's going to be my temperature. In fact, again, I might just, to make life easier, I'm going to copy and paste it straight from my table heading. So that's done. Now, personal preference, really how you like your axis label. You can leave it on the side there so it takes up a bit less space, or you can make it horizontal, which is what you often see in exams if that's easier to read. Things are important here. It's really not relevant to have the temperature right down to 0. So if you double click on this axis, it brings up a few options for formatting on the side. So at the moment we're in axis options, which gives us all our choices to do like mathematical functions or graph functions. Text options is more to do with like how it looks in terms of size and color and stuff. So axis options, I'm going to make my minimum here, I think 30 is reasonable, and it goes up, that's probably fine then, up to about 55 degrees, spans the increments we've got. Now you can change it so that it goes from like 32 to 52 if you want, and have those matching the ones you actually measured if you prefer, so that the dots fall on the major axis lines. That's one way to do it, which would be quite suitable. Or if you want, because we've picked sort of slightly odd increments because you wanted things around human body temperature, we can go back to this range that's sort of on the 5. As long as you've got grid lines here that pass through your actual data points so that somebody reading this can see that that is very clearly 32 degrees by reading down here. Either is fine as long as it's clear that your data points can be read off your axes. So we've got here this range, and we can see because this actually isn't human catalase, it does actually function optimally a little bit higher, but it's hard to pick what our optimum is. We might say it's this 42-degree one here, although these points are quite similar. So this is where our error bars are going to come in to show us if we've got any overlap here in our values, to say with confidence or not, you know, where sort of our peak of our graph is. So it gives us a bit more information. So click here on this plus chart elements again to add more information. I'm going to click error bars. Now you'll get this nice big mess, so that definitely needs some extra work. So let's go to more options down here. Unfortunately, it puts both on, which we don't need. We don't need these Y ones, these horizontal, X ones, sorry, these horizontal ones. So if you click on them, so just those ones come up, see these little blue circles, they should only be on the horizontal ones, the X error bars, and just press delete, and they go away, because we're not looking at any big dramatic range in our temperature values. Now, Y error bars up here, if we go to our error bar options, click the little graph button here. So we want those in both directions. We're going to show the variation above and below our average. Now here it's done standard error, which is a bit like standard deviation, just a little bit smaller, divided by the sample size again. But unfortunately, if you pick one of these ones like standard deviation or standard error, that's the standard deviation for the entire data set, not each temperature value. So that's where we do custom, and we specify the value, and we click here. Positive and negative means how big the error bar is above and below your data point. So delete what's there, and then have a look here, just click your standard deviation values that you calculated. So you pop them in for the positive value, and then you do the same thing. Delete what's there for negative and highlight the standard deviation values and click OK. So now we can see, we should see that the biggest error bar is our biggest standard deviation value. So if we looked at our data, they're the ones that have the biggest variation amongst our trials here. So we could say then there's actually quite a bit of overlap between our error bars, between these data points here through the middle. So we can't be confident about saying that this is our optimum. What we could just comment on is that the optimum temperature for this enzyme is perhaps within this approximate range. But we could see here, we could say we're quite confident that it's significantly denatured or less active at this higher temperature value, because that data point is very clearly removed from the others, and the error bar, there's no overlap at all between those ones. So that's an example of how we might work with some data here for our enzyme experiment, and just generally some tips about making a graph and presenting data for an IA in biology. I will just quickly show you another option too. Some of you worked with values, two values for your data collection. So there's just a little extra step of processing there. So let me show you one where I've put in lots of data already. Here's one I've prepared earlier. Okay, so the numbers might look a little bit overwhelming at first. Don't be alarmed. This is a sample, just some mock data. If we tested the effective pH on catalase, I've just picked a different variable this time. And this is one if we do mass loss, because if you measured mass loss, then you would actually have two values. You would have the initial and you would have the final mass values, and you would need to report both in your report. So here in our table, we would have the mass. We would indicate that that's at the start time and 30 seconds later. Again, we would have our values. We would have an uncertainty, plus or minus. Now that would be for digital scales or any digital instrument. That is the smallest increment we put as our uncertainty value. So if our scale's read to 3 decimal places, that's our uncertainty. And then again, we've got our trials with catalase and we've got our control. And we can see we've got starting mass and end mass. So they've gone down here. Now that's the start with the, and as we've indicated, the mass of the whole flask and the reactants. So we've got these values that would be our raw data or unprocessed that we collected our results during the experiment. And then we would have to do some calculations here where we look at the difference. So we take our start value and our end value and subtract one from the other. And then we're still doing it as a rate though. So if this is again over 30 seconds, we'd have to divide our answer by 30 to get a rate per second. And again, for our per second, we're using that -1 subscript here, that index. I've just put a little note here to remind myself to tell you as well, with significant figures, when you do adding and subtracting, like here, if we take the difference between, say, this one, 144.552 and 144.547, these values have 6 significant figures. But when you find the difference, you know, the difference is quite small here. Don't go trying to fabricate extra significant figures. You just keep the number of decimal places consistent when you're adding and subtracting. So a lot of these, that one would only have 2 significant figures, but actually a lot of these will have about 3. So just watch out. Don't get carried away going to a million decimal places here. When you're adding and subtracting, just work with the number of decimal places that were in the data you collected. So looking down here, what I've done, the formula I've used, if we look at this formula bar and it highlights the cells it takes information from. So for the first trial, I've done the difference, B5, the starting mass, subtract C5, the end mass, because we had a mass loss. And then I've put them in brackets, otherwise BODMAS gets in the road or it will just divide one value by 30 instead of the difference. So I've put brackets around that information. I've used the equal sign to tell Excel I want it to do the calculation. So just equals, bracket, the cell with the information, B5, press the hyphen for the subtract, and then click C5 for the other cell. Close brackets, and then / is the division symbol in Excel, and by 30. And if I press enter, we get the result. So that's my rate of mass loss per second. Now, this one, annoyingly, if you try to copy that formula to the next cell, it will stuff up. Watch what happens. Control, if I just drag it, click and drag, and then have a look at the data. It's moved because my value here in the results goes across by 1 to the right. It also adjusts this information that the formula is generated with 1 to the right as well, which isn't enough. We actually need to go 2 across because we've got the start and end data. And unfortunately, you've just got to go through and manually fix that. So you've got to go and change that to D5 and E5. So that this one lines up with these 2 values, this one lines up with these 2, this one with these 2, and so on. Okay, a little bit tedious, but not the end of the world. If you fix it in this first row, then you can highlight that first row and copy and paste them down, and it's fine. What have I done something? Oh, sorry, because I've actually gone and changed. If they're all right. Make sure that they're all consistent. I've just fixed the decimal places. That's why they came up a little differently there, because I've already, as a shortcut to making sure this video doesn't go on forever, I've already adjusted the number of decimal places so that they have the right number of significant figures. So I've gone through, I've got my start minus the end mass divided by 30 for the change in 30 seconds. I've got a mean, my equals average formula, and I've got my standard deviation equals STDEV, again with brackets and highlighting the information. This one, I did have a little bit of mass loss with my controls, slight bit of evaporation or a tiny bit of degradation. So I have converted those into a rate, but they're so tiny that they're practically negligible. But it's worth discussing, because even the ones, say, where the enzyme looks like it's not really working, but perhaps a tiny bit, you can compare and say, well, you'd expect to get a very minute amount of mass loss anyway in an open flask on a set of scales. Now, these values here are messy. They look horrible with all these decimal places and zeros. So it would be much better and following convention to put it into scientific notation. Excel can do this for you. If you click on the button and up here it says number, you can actually put it into scientific notation like this. It's still a bit messy though, because Excel puts this E, means exponent, puts a capital E and then minus 0.4. So what that actually means is times 10 to the minus 4, which is okay, but it's not really convention to use an E. That's just Excel's format because it doesn't put a little time symbol in there. And it also means that all going to be different. Some of them are going to be 10 to the minus 4 and some of them are going to be 10 to the minus 3, which is not as good for direct comparison. What I suggest you do is have a look at your biggest values. So the biggest values here, 0.009, around those ones. That would be, if you put that in scientific notation, they would be times 10 to the power of minus 3. So I recommend you make them all that. And actually, what the better thing to do is to copy the values in this table or change the formula so that you also put in here asterisk, which is times, multiplication in Excel, times 1,000. And then in your units, put a little times 10 to the minus 3 font superscript. So if you change all of them, here's one I've done earlier, don't look as horrible anymore. So these are in scientific notation and they're all times 10 to the power of minus 3. So I can put that in my heading. And it will also just look a bit less messy on your graph. So these are my values for rate because mass losses are really small. My trials are catalase, my control, my mean and standard deviation. And again, what am I plotting? Here's my graph that I prepared earlier. So my graphs here, I've deleted the title that Excel generates automatically and I've put my own figure 1, the effective pH, blah, blah, blah. I've got my pH here because my independent variable is my x-axis. I've got my rate of mass loss for my enzyme catalyzed reaction. I've got my grid lines, extra grid lines on there so I can read a bit more of it. This one was suitable to use a smooth curve. So when I went to my chart type, I selected an XY scatter and I did the data points with the smooth lines. I've also, this one was quite wordy. So in the axis titles options, if you like the look of it, here I've got my text options. And then here, this text box gives me my direct text direction. And I've put that to horizontal rather than the automatic like vertical or stacked one that it will do by default. So I've just selected horizontal for that one. Otherwise, all the same things. I haven't bothered adjusting this axis minimum and maximum. It is actually possible to have a pH of 0, though usually we deal with a scale from 1 to 14. You can adjust that if you like. Again, just double click or go to the extra options if you want to make the minimum maybe just 2. That's suitable. It's up to you. Just getting rid of excess information here. The y-axis will automatically use the maximum number of decimal places in your data. So you don't have to change that one. The grid lines, make sure here with your, if you click on those grid lines, so that means your like background graph and it brings it up with blue points. In fact, oh sorry, actually it's actually on axis here, if I go back to the numbers here and go to access options on my little bar graph picture, make sure that the major units are 1 if they're not already, because you want all the pH values on there. And I wouldn't go any smaller than 0.5. That's not necessary for the other smaller lines on your background graph paper, because you're not really reading to your pH values to tiny degrees here. I think that's all the main things, but please ask me if you've still got some questions. Hopefully that's a good pointer too, or when you're doing any prac, to go back and look at what's a good way to set up your tables. Make sure you've got your independent value here, independent variable, sorry, so your increments that you're testing down the side here should be at the start somewhere here on the left. You've got all your trials, you end up showing what your mean is, that your standard deviation, oh sorry, I didn't put error bars on this one yet, that you use those values as well. So error bars, tidy them up, get rid of those horizontal ones. The Y ones, double click or click more options. So you can go down to custom, specify the values using your standard deviation that you've calculated. And one last thing actually to point out, these ones where I changed it to scientific notation times 10 to the minus 3, because I made that times 10 to the minus 3, I multiplied them by 1,000, which is 10 to the 3. When I do the average, I don't need to put that in there because it's taking an average and standard deviation of values that have already been multiplied by 1,000. So I don't need to do that again to the mean values. If I did that times 1,000, they would be really thrown out. You just messed up my graph. So you don't need to do that twice. So just have a look. When you're working through Excel, you'll see if you've stuffed up, you'll get something really strange like that on your graph. Have a look, or if you've subtracted the wrong thing, if you did the final mass minus the start mass instead of the other way around, you get negative values and you realize straight away that you've done something wrong. So make sure you go back, have a look at those formulae and that they make sense, check they all work. Sometimes you just left off an equal sign or something if it's having trouble calculating it. Or if you're still having trouble with Excel, just write a normal data table. Just do it in Word and use your calculator. That's fine. Since Excel is quite convenient because once you do get a whole row of formulae correct, you can copy and paste it and it actually ends up saving you quite a lot of time. I think that's it. There's some sample graphs. Good luck with your own ones. Again, pop a comment on here or just contact me directly if you would like some help with your data tables as well.